HOPTEC project or program. So as you probably already know, this was a collaboration launched last year between Hopkins and Technical, and Andre and I have been working together to keep the program going. This year we had 2 new students who have been developing their master's work at Hopkins, and I think Ines is here, and Alfredo will probably join. And we think it has been a great success so far. Stay tuned. We're going to have a symposium coming up at the end of the year. And our official website will be launched hopefully by next week, so you can see all the updates. And without further ado, Andre, I think maybe we can get started with introducing the speakers, right?
Yes. So Dr. Phillip is an assistant professor in the Department of Biomedical Engineering with additional appointments in chemical and biomolecular engineering and ecology. He's also a core member of the Institute for Nanobiotechnology, where his interdisciplinary research bridges engineering, biology, and computational methods. His lab integrates fundamental engineering principles with medicine and machine learning to explore how cells encode information related to aging. Notably, his research focuses on understanding why individuals age differently and developing strategies to promote healthy aging and combating disease. Without further ado, thank you for joining us, Professor Phillip. It's a pleasure to have you. I just want to note that I saw you walking into the wet lab 2 minutes before this talk, and that's, to me, amazing how you can manage both things. And the floor is yours.
Yeah, thank you so much for the introduction and for the opportunity to speak a little bit about our work. If you can give me permission to share.
It should be okay now.
Yep, works now. All right. So today I'll speak a little bit about some of the work that we've been doing in my lab on the topic aging and senescence at single cell resolution. And just to confirm, you've seen one screen, right? Not the two. All right, perfect. So the demographics of our populations are changing drastically, right? So did you know that by the year 2050, about 1 in 5 individuals would be above the age of 65? And this statistic is particularly for the US, but it's similar across the different countries of the world. And really, we're heading towards this changing demographic from really 2 streams of changes, right? So one is this older population is increasing because healthcare infrastructure is getting better. So some of the things that people were dying from, such as infections and so forth, are not as common. But then the second stream is the fact that there's declining birth rates across the world, right? Particularly, projections show that by the year 2040, there'll be more people above the age of 60 than there are below the age of 18, right? And again, that statistic is particularly for the US, but then there's similar trends that are seen across the world, right? So in essence, this changing population or this changing demographics is set to change how our society sort of runs from the perspective of like socioeconomic burdens and also from the perspective of health. So while I think it's a great thing that individuals are living longer, one of the things that we don't think about as often is the fact that we're living longer doesn't mean we're living healthier, right? And as I just mentioned, what that does is it tips the balance away from health towards these conditions where individuals have multiple diseases, which we call multimorbidities. And as a result of these multiple diseases, they're taking multiple different types of medications, right? So which is called polypharmacy. And we know how expensive medications are. So in essence, this shifting demographics is shifting away from health, but the implications go so much more than just the individual in terms of our individual health. But as engineers, I think, we can look at this and see challenges, but there's many different opportunities that we can really innovate on top of, right? To be able to engineer better health or engineer more resilience within our population. But contrary to what we may hear in the media, I don't think the problem is why are we aging, right? Because when we think of it, aging is a natural process, right? It's a process that takes, every year we gain 1 year, but our health doesn't necessarily match with this changing in year. But I think rather the key problem or the key question we should be thinking about is, why do we age differently? And thinking of it from the perspective of health, right? So why is it that some people are more prone or predisposed to certain types of diseases, whereas others are less so, right? And one way we can think of this is by, using this illustration that I have down here, in the sense that we can look at groups of 80-year-old individuals, right? And we can find some 80-year-old individuals who, are very fit, are very strong, very healthy, and they can run marathons, right? Whereas we have other 80-year-old individuals that need a lot more assistance, need a lot more help. And then you have people in the spectrum in between. So the question begs, why do we see such a range of biological differences or health outcome differences, even though they're all chronologically 80 years old? And what that leads us to is that there's a difference between what we can think of as our chronological age, which is, in essence, the time since we were born, right? Every birthday we gain 1 year, versus what we can think of as biological aging, which starts to tell us more about how our biology is changing over time, and then how these changes in biology really impact our overall health. So in my lab, we try to frame it this way, in the sense that there's this difference between chronological age and biological age. But really, we need to better understand the biological aging process so that we can then impact disease, right? Or impact the overall health of individuals. So we can sort of like think of it through this phase diagram, where on the x-axis, you have chronological age, right? So it's just a linear increase in time. And on the y-axis, you have what we can think of as health outcomes or overall health, right? And in healthy individuals, right, which I imagine that all of us, strive to be healthy, we fall along this green line, right? In essence, you have a certain level of health, which is, we can think of it as good health. And that health declines over time, but it declines very slowly. But on the other hand, we can have individuals who have maybe predispositions or certain types of ailments or diseases that this decline is very fast, right? And then you can think of every possibility in between, which represents, all of us in the world or so. But the question is, how can we effectively gauge this decline? And are there ways that we can understand it better, that we can then create interventions that can promote healthy aging? And to do that, the fundamental question is, can we modify our biological aging trajectory? Because I think we can all agree that we cannot modify our chronological aging trajectory, because time is linear, right? We're just going through time. But the biology that's associated, can we actually modify that? And in order to modify it, we need to be able to measure things, but we need to measure things that matter, right? So there's many things that change as we age. So for instance, we can look at different people, different ages, and there's a change in the number of gray hairs, or the loss of hair, or number of wrinkles, and things of the sort. But I would argue, from a biological perspective, the amount of gray hair that a person has tells us very little about their biology. So the question becomes, what can we measure? Is it actionable? And how can we actually use that information to go forward? So in my lab, we think that cells provide this window into health that gives us a preface of what is to come, in the sense that before diseases manifest at the physiological level, they first start as changes in cells. We can think of those examples, for instance, like cancer, right? Before you get a full cancer mass, like, it starts off as dysfunctional single cells that then propagates into this disease. So we're looking at single cells, whether using human samples or in some cases animal models, and we do this deep phenotyping on it, particularly in terms of like looking at dynamics and how their behaviors change in a variety of different biological contexts. with the hope that we can develop these systems of information that we can define individuals and then essentially inform on their health and different health interventions, given the changes that happen over time across these different scales. So over the years, there's been many studies that really tries to map out what are some of the things that change as we age, right? And one of the areas that we've been sort of fascinated with is this notion of cellular senescence. And here I'm just showing this wheel, hallmarks of aging, that tries to explain some of the drivers of aging from a different across different scales as well. So cellular senescence is mainly defined on the cell scale, but then you can have different things, changes at the genomic scales and so forth. So senescence is really identified driver of aging and aging dysfunction, and it's conventionally described as this state of cell cycle arrest, where the cells move from this proliferative state to a non-proliferative state. They bypass apoptosis, and as a result, they start secreting these pro-inflammatory factors that we call SASP, for senescence-associated secretory phenotype. And interestingly, these cells also exhibit very strong morphological changes when cultured in vitro, right? They tend to get very large, and they tend to be, you can tell the difference between senescent versus non-senescent just qualitatively by eye. But interestingly, one of the fascinating things about senescent is the fact that it is a cellular phenotype. So it's defined at the cell scale, right? These cells are changing, but they have long-ranging implications in the overall physiologies of individuals, right? So senescence has been associated with aging, so they accumulate with aging and a variety of other diseases, but they've also been implicated to have beneficial roles in things like wound healing and development and so forth. More recently, there's been a new class of drugs called xenotherapies, which are mainly repurposed drugs, many of them repurposed cancer drugs, that has shown to be very effective in modulating aging-related dysfunctions by targeting senescence. So for instance, there's a series of papers over the last 10 years or so that has taken these animal models that enhance senescence, so it's like P16 ink attack models, and then you can see the dysfunction that happens with age. So these mice become like very hunched, they get weak, their fur becomes very white, and they look frail. And then when you eradicate or deplete these senescent cells, whether genetically or using drugs, you get this reversal of a phenotype, right? So the mice again start running around, they get a lot of energy, their fur becomes shiny and black again. So there is this change that happens physiologically when you actually target senescent cells. But the question that's been in the field for a while that we don't fully understand is, when is senescence good and when is senescence bad, right? Because there's many papers that show senescence, if you eradicate or deplete senescent cells, you get a beneficial effect. But then there's also papers showing that if you remove senescent cells at the wrong times, for instance, during development or during wound healing, you actually make things worse, right? So you actually need the senescent program to actually run to orchestrate normal physiology, but then at the same time, you don't want it to be lingering so that it causes long-term effects, particularly during aging. So in the field, we sought off to sort of like look at this problem and paying particular attention to these two big gaps that are in the field. One being there's no universal biomarkers of senescence. So currently we use a handful of protein-based biomarkers, so P16, P21, lamin B1, and so forth, but none of them individually identify senescent cells in all contexts, right? So it's very context dependent, and these biomarkers are proxies, but they're not perfect. And then secondly, we know that senescent cells accumulate with age, but it's unclear whether aging cells are just more prone to becoming senescent or it's really just a clearance issue where we're unable to clear these senescent cells effectively during aging. So our approach to this was, in essence, to not think of senescence as a binary phenotype, right? Right now, the field thinks of it as a binary phenotype, meaning a cell is either senescent or non-senescent. And as a result, the context dependence is a bit ambiguous. So our goal was to identify and classify what we call functional subtypes of senescence, because our notion is that within any cell population, you would have different senescent cells, and these senescent cells come in different flavors, which we call subtypes, that may have different functional roles. So to do this, we're going to combine experimental approaches, meaning high content imaging and microscopy, with data science and machine learning to be able to develop this unified approach based particularly on cell morphologies. And we're not the first to actually use cell morphologies to identify senescent cells. There's 4 papers that have come out in the last 3 years that have really essentially demonstrated proof of concept that it can work. However, in each of these papers, there's been assumptions that have been made that really doesn't allow for the investigation of functional subtypes. Mainly that any cells exposed to senescence inducers, which are mainly chemotherapy drugs, are senescent, right? And this blanket statement doesn't allow us to look at subtypes, because then you have these mixtures of populations that you then train models on. So our model is a bit different, where instead of using 1 inducer, we use 5 inducers. Instead of using 1 biomarker, we use 5 biomarkers as well. And then we build out this whole computational framework and analysis pipeline using cell morphologies as well as multiple protein-based biomarkers. So this is just a bit of the information in terms of like how we're actually doing it. So this is an example of cells at baseline. So these are dermal fibroblasts taken from a healthy 23-year-old. And then these are the same cells induced with hydrogen peroxide after 8 days, right? So you see a drastic change, both in terms of the cell morphology and in terms of the nuclear morphology. And a little bit more detail in how we're inducing these cells. We're using hydrogen peroxide as a reactive oxygen species induction method. We're using bleomycin and doxorubicin as a DNA damage response. We did replicative in terms of just serially passaging these cells, as well as proteotoxic stress using this ATV drug, which is currently used for HIV treatment. And then in terms of the biomarkers, typically most studies use P16 or beta-galactosidase. We're using all 5 of them, multiplexing them 2 at a time, since we are limited in terms of how many channels we can do. But so beta-galactosidase, P16, P21, HMGB1, and lamin B1. So as a proof of concept, this is just some microscopy images showing the same 23 cells from the same 23-year-old person. These are untreated, so these are baseline. And we can see essentially the 5 biomarkers in addition to actin that we use for the cell boundaries and DAPI that we use for the nuclear boundaries. And then upon induction of senescence, these markers increase, right? So beta-gal, P16 and P21 supposed to increase based on literature, and we also see this increase here. HMGB1 and lamin B1 are supposed to decrease based on literature, and we also see that decreasing. So this is essentially our sanity check, our proof of concept that what we expect to see given the literature is actually what we're seeing in terms of the rough expressions of these different biomarkers. And here we're just showing that if you look at these different biomarkers across the different induction methods, overall, you see the trend where when they're supposed to increase, they increase. However, when you start looking at the actual magnitude of the expression, you see that it is induction dependent, right? So the way in which we induce these cells actually influences the magnitude of the expression of these biomarkers. So that was a proof of concept. So in terms of our workflow to really identify these functional subtypes, we use image processing. So This is a field of these fibroblast cells stained for actin and nuclei. We show that we can do segmentation on the cell bodies and the nuclear bodies. And then from this, we're asking, are some of the features that we use to define the cells or features we use to define the nuclei, are they associated with these senescent-based biomarkers? And then if they are, can we use morphology as this proxy for senescence that takes into account this multi-dimensional, in a sense, type of view? So to do this, we use, we're measuring, with each of these cells, we're measuring hundreds of different features, right? So in order to really visualize it and interpret, we use reduced dimensionality. Here, we're just showing it based on UMAP. And each dot here is a single cell, right? So these cells represent the 23-year-old, which we call young, and an 89-year-old, which we call old, right? So you see that they localize within, the lower left of the manifold. And then on top of that, we can layer in all the cells across all the different conditions, the 5 induction conditions that I mentioned before. So this is when we add in cells induced with ATV after 8 days, and we see these cells are more located to the right. We can add in the replicative senescence, doxorubicin induced, bleomycin induced, and hydrogen peroxide induced. And with that, we fill out this whole space within the UMAP. And roughly what this is telling us that there seems to be a polarization of this morphological progression towards senescence in this manifold from lower left towards the right. So can we actually validate this? So when we can also do k-means clustering on this UMAP space, and we get 11 different groups. And these 11 different groups are defined distinctly based on morphologies, right? So what we see is on the left, these are, smaller cells, more fibroblastic cells. On the right, these are all like very large looking fibroblastic cells. And everything is to scale. So the actual change in the size you're seeing is an actual real change. And then we can layer in the different types of information that we use to define this space, such as nuclear area, cell area, and we see, nuclear area cells. Nuclei are larger on the right, cells are larger on the right, which is qualitatively what we see here. So in essence, we see a size polarization from left to right in terms of increasing size from left to right, and then a shape polarization in terms of lower aspect ratios on the top, higher aspect ratios towards the bottom. So a top-bottom polarization that defines really the size, the shape. So from this, we can actually go back and ask the representation of each of these clusters within the different, the young and the old population that we looked at. And we can get these fingerprint profiles where at baseline, you have very low abundance of these clusters over here, these like 7, 8, 9, 10, 11. Whereas upon senescence induction, you start seeing there's an increase in the abundance of these and less in the abundance of the earlier ones. And I'll get a little bit deeper into it as we go. We can also classify senescence in terms of the biomarkers, and it shows what we expect, that these morphological changes are associated with the changes in the biomarkers as we would expect. So if we were to classify non-senescent and senescent based on these biomarkers, we see the non-senescent ones are on the left, the senescent ones are on the right. However, if you look at the contour maps of these 5 conventionally used biomarkers, you can see that the localization of the high expressing or low expressing cells are somewhat shifted, right? Meaning, especially on the cells on the borders, that in one biomarker, they may be thought of as senescent or classified as senescent, whereas in others, they may be classified as non-senescent. So having all 5 of them actually gives us an advantage in terms of that. As a second way to kind of like test this, the first way was all parameterization. So we defined all the features. The second way is we asked if we can build a model that then just uses the raw images to be able to ascribe a senescence score for each individual cell. And then we can compare the two to see if they sort of like coalesce onto the same result. So we use the exception architecture, and then for each of these, we either input the nuclei from the DAPI channel, the cell from the actin channel, or the composite. And then we can ascribe essentially a senescence score where 1 is senescent, 0 is non-senescent, and they're all probabilities, right? So if something is 0.8, there's an 80% probability that it is senescent. And the model works really well. We have over 89% accuracy in our validation and testing set. And if we were to paint on the senescence scores on the same UMAP, we again see the shift towards high probabilities of senescence on the right versus on the left. And then we identify these 3, what we call bona fide senescent subtypes, right? Which they have an average score of 0.98, 0.98, and 0.91, which we then call cluster 7, 10, and 11. So having identified these across 2 different sort of orthogonal approaches, the question is, are they functionally different? And we think they are. And I'll show you 2 lines of evidence just to show that they are. But in essence, what I've shown so far is that the model can predict senescence at a single cell level, right? So this is not saying this population of cells are senescent or that population of cells is non-senescent. We can actually ascribe this senescence probability at a single cell level. And that allows us to ask deeper questions in terms of likelihood or susceptibility towards senescence and so forth. So the first line of evidence that we think that they are functional is the fact that they change differently with age, right? So if you read any senescence paper, they'll tell you senescent cells accumulate as you age. So we took a cohort of 60 people. We profiled their senescence burden at baseline, and we see there's an increase in senescence as you increase age. However, if you now break them down, given cluster 7, 10, and 11, you see that 7 doesn't really change. The correlation is like, there's no correlation here with 7. There's a strong, decent correlation with 10, and a weaker but still positive correlation with 11, right? So I would argue if we're trying to target age-associated senescence, we probably want to target 10. So that's one line of evidence to say that they're potentially functional. And the second line is, do they respond differently to drugs? So we took these signal-therapies, D+Q, which is a drug combination in clinical trials, and we exposed senescent cells to them on the microscope across 3 days. So in essence, we induce these cells, wait 7 days, put them on the microscope, expose them to drugs, and then start imaging every 30 minutes for 3 days to be able to follow the death or the response of every cell within our field of view over time. And what we see is that, there are some fluxes with time, but 10 is they're pretty stable in the untreated, but in a D+Q, we start seeing death. And as a bulk population, 7 seems to be most susceptible to D+Q, and 10 seems to be most resilient to D+Q. And putting it a different way, we can classify the responses. So this is just linear time. When we see black, it means the cell is dead, no longer there. And we see these 4 responses. These where they die, these where they don't die. And we call this long death, short death, stable viable, and unstable viable. Meaning that, if you're purple, you stay purple. But here you kind of fluctuate between the different groups. And overall, we see, as we expect, in the death cases, there's more in the D+Q because we're actually trying to kill them. But then if you look at the responses as a function of subtype, you see that in the 7 is most susceptible overall to death, and 10 is most resilient, meaning these are the ones that a higher fraction of them stay viable over time. So we're really excited about these results, and we're excited to push it forward. So what I hope that I've been able to demonstrate is that senescence is not a binary phenotype, but there exist these multiple subtypes. And currently what we're doing is trying to get a deeper understanding of what are the molecular drivers of these subtypes, be it epigenomic, genomic, or metabolic, and how does that apply and give us new ways to find senescent cells in tissue. as well as we want to gain a mechanistic understanding of how context matters, right? The context specificity of it. And then lastly, by understanding these molecular drivers, we can then identify molecular targets that allows us to target one subtype versus the other, right? Which is, for instance, what I showed, that if you want to potentially target the age-associated one, you probably want to do 10 as opposed to 7. So we think it matters because understanding all of these, particularly a driver like senescent, is important in understanding overall health. And what we really want to do is to be able to understand how we can move people from these accelerated aging trajectories towards something that's slower, that then we can essentially gain health, right? So to preserve health as opposed to just, reversing aging. And with that, I'll stop here. And thanks to my lab for all the work. So all the senescence work was led by Pratik Kamat, who is a graduate student in my lab, with help from a number of different graduate students in the group, including Nico. And with that, I can take any questions if we still have time.
Thank you so much. That was amazing. It always strikes me how you can extract so much information out of just nuclear morphology or cell morphology in this case. I'm going to open the floor for questions. I also have some questions of my own, but maybe we can start with some people from the audience.
I could ask you this. I don't know if I'm remember.
Okay, we can hear you now.
Jude, I think it's so cool how you can map senescence in such complex ways. Do you think any of this could apply to identifying senescent cells in tissue as opposed to in a dish?
Yeah, so we think the in vitro way allows us to distinguish the subtypes, right? But then we need to some ways like map the information in the dish to the information in tissue. So we like to call it, the zeroth law of thermodynamics, where if A is B and B is C, then A is C. So we're trying to use molecular features like gene signatures or epigenomic signatures to identify the subtypes and map them, and then go back into tissue and use these signatures to identify the different subtypes. Because there's some interesting data and there's a lot of variability in terms of identifying senescent cells in tissue. And we think part of it may be due to the mechanics of the senescent context. So we've done some in vitro studies and shown that none of the biomarkers work if the cells are in a soft context. However, the transcriptional signatures turn on. So the transcriptional programs work, but the protein-based ones don't. So then that gives us a little bit more confidence that using these molecular signatures might allow us to do it better in tissue.
Vasco, go ahead.
First of all, thank you so much for the amazing talk. Like Andre said, it's just astonishing how much information you can get out of these images. My question is a little bit more of a technical one. For the exception model you trained, what was the ground truth for what is a senescent cell and what isn't a senescent cell? Because I'm guessing even if you treat cells with the senescent-inducing drugs, some of them will not be senescent, some of them will. So I was wondering, how were you able to train the model?
Yeah, so that's a good question, right? So the way that we did it was partly because we have 5 of these different biomarkers. So what I didn't show was that you could only image 2 at a time, so P16 or P21 and so forth. So we multiplexed it and we built like an imputation model where we can get the 5 markers in every cell. So the way that we identify the ground truth of senescence is by saying you have to be positive or identified as senescent given 2 or more biomarkers. Yeah.
Okay.
That's like one way to do it. I don't think it's a perfect way, but as we learn more, we're kind of going to get better at what the ground truth is.
Thank you very much.
Okay. Any other questions? Okay. For the sake of time, we'll move forward. I actually had a couple of questions, but we'll catch up later. So our next speaker is Professor Cláudia Lobato Silva. She's an associate professor at Instituto Superior Técnico. I had the pleasure of having her as one of my professors back then. And so she received her diploma in chemical engineering from Instituto Superior Técnico as well back in 2001. And then she also received her PhD in 2006 in the same institution, but at this time in collaboration with University of Nevada, Reno, USA. Her research focus, within the stem cell engineering research group of IBD, includes expansion of human stem cells, cellular therapies with human adult stem cells, and bioreactors for stem cell culture. So we're going to have a little bit of a stem cell lecture now. Thank you so much for accepting, and the floor is yours.
Thank you so much, Ines. Are you hearing me okay? Yeah. And first of all, thank you for the kind invitation to these great ambassadors who are Inês and André, but also the newcomers. So, right, we have Vasco, Inês, Alfredo here. So it's really a great pleasure to join you in this session today. So I come from an engineering background, chemical engineering specifically, but I have been working on stem cell technology. So we are part of a research unit in Portugal, which is Institute for Bioengineering and Bioscience. And this is the Stem Cell Engineering Research Group in which I coordinate this focus area of development and manufacturing of cell and molecular therapies. So basically what we do, we work on identifying signaling mechanism under specific cell microenvironment, mostly related with stem cells, but not only. Then we aim to design and establish processes for the manufacturing of these cells for their clinical use. And at the end of the day, we aim at the translation of these processes and these new products towards the development of the so-called advanced therapy medicinal products. And we saw a great lecture now, the previous presentation of senescence and the onset of many diseases. And for many of these diseases, we do not have a really great offer from the therapeutic standpoint. And there is this new category of advanced products based on cells. I have been mostly focused on cells to tackle these unmet medical needs. So over the years, we have been working hard on different tissues. Okay, so in this great cartoon produced by one of our talented PhD students, we have here some examples of the primary tissues and fluids that we work with: the umbilical cord, the bone marrow, the adipose tissue, even more recently the bladder, the muscle. And we optimize methods for the isolation of specific cell types. Then at the second level, here in the middle, you see some of the platforms and the technologies that we use to multiply the number of cells available. Most of these interesting cells are really scarce and limited in cell numbers. So we use technologies to amplify the number of cells available for clinical use. And more recently, we are also interested, besides the cells, some key products produced by those cells, such as the secretome. We saw some things also related with the secretory capacity of the cells in the previous talk. And also the extracellular matrix. So lots of things going on about the cell-based therapies. And when we are trying to design and trying to develop a new process or identify a new cell product, we face lots of challenges, mostly related with the clinical side, okay, and we are not clinicians, but some of them are really engineering challenges. One that is my favorite is the scalability challenge, okay. For most of these applications, we have very interesting cells, but they do not have a clinically relevant dose. So you have to multiply the number, hopefully keeping the cell potency and the function, but it's really a critical challenge. Then all the ingredients that you use to cultivate activate these cells should be able to comply with the clinical use that is intended for these cells. You have to, with the clinicians, of course, and with the biological science underlying all of these, you have to decide whether or not you should use cells from the patient or from a donor. A very interesting point that we have been working on is about how to store the cells. So cryopreservation is really popular. Everybody cryopreserve the cells, but are the cells really great after cryopreservation or not? So we have been working on some of these challenges. And one of the population of cells that we have been mostly interested is these mesenchymal stem stromal cells. In some literature appears as stem, others more stromal cells, but these are very interesting cells because they do differentiate into many different tissue types of our body that everybody, even if it is not a big fan of stem cells, identify as a very important tissue: bone, cartilage, tendon. And also one of my favorites, so these are the cells that give rise to the marrow, which is the inner cell part of our large bones that support the blood cell production in our body. So these mesenchymal stromal cells are the ones that support the blood stem cells to produce red blood cells, platelets, and white blood cells. And there is lots of fuzz about these cells. They are really popular, lots of things going on, but also lots of controversies. Okay, so this was the cover of Nature in 2018. Basically claiming, let's clear up this stem cell mess, because probably the enthusiasm of mesenchymal stromal cells for clinical use went too fast, okay? But still, there is lots of great data from clinical studies, clinical trials, using these cells, mostly from the bone marrow, so from the traditional source, which is a bit invasive. So lots of clinical studies are seeking for alternative ways to obtain these cells. The adipose tissue is an alternative. Also, some cells of these characteristics are also found in neonatal tissues, such as the placenta or the umbilical cord. And we have been working on many of these alternative tissues for obtaining these mesenchymal stromal cells. And in the clinical studies that you see, you can see a set of studies targeting the differentiation of these cells, okay? So mesenchymal stromal cells give rise to bone and cartilage, so you can think of treating conditions affecting those cells. But most of the clinical trials, interestingly, they target a different way of regeneration. So rather than relying on differentiation, these cells can play with immunomodulation, can secrete factors that can be very pro-regeneration. And not surprisingly, lots of clinical trials targeting autoimmune diseases such as rheumatoid arthritis, Crohn's disease, and also some acute conditions such as myocardial infarction. And in Europe, not still in US, but in Europe, we do have a product already approved in the market based on mesenchymal stromal cells. It was approved quite recently, in 2018. It is used to treat very specific and complex fistulae in patients with Crohn's disease. Crohn's disease is one of the examples that I just gave, and it is an inflammatory condition of the gut. And a very particular set of patients do have very complex fistulae that do not respond to any treatment, okay? So these are the patients that can be eligible for these innovative therapy of mesenchymal stromal cells. However, if we take a look onto the manufacturing of these cells, we do see lots of limitations of the process that the pharmaceutical company that produces these have. So mainly the process still uses very undefined reagents, fetal bovine serum. So all of you here in the audience that already had some cell culture do know that fetal bovine serum is quite popular, okay? But fetal bovine serum has lots of limitations related with, for instance, the allophysal, this product based on mesenchymal stromal cells, come with this alert, which is it should not be given to patients with allergy to bovine serum. And this is because the cells are produced with this undefined reagent, which is a source of contamination, and it can also provoke some immunological reactions upon the administration of the cells. So this is one of the key limitations of the manufacturing process of the product that is already in the market. The other downside and technical limitation of the manufacturing process is that it's really based on the conventional method of using plasticware, those Petri dishes or big flasks. So these cells do attach to the bottom of the flask. So they are really sticky to polystyrene that we use in these flasks. And basically you have to use tons of flasks, lots of incubators to have the cells that you need for clinical implementation. So here, definitely we saw an opportunity to, okay, what about using more sophisticated ways to manufacture these cells? Namely bioreactors, okay, for those of you, and some of you have a biological engineering background, you readily recognize bioreactors that have been used for decades in the pharmaceutical sector, for the production of vaccines, for the production of lots of biologicals. So the idea would be, okay, whether it's possible to implement a bioreactor process for the manufacturing of stem cells in general, and for these mesenchymal stromal cells in particular. And when we decide to implement a bioprocess based on bioreactors, firstly, we have to think, okay, so these are my cells. The cells typically attach to the plastic, so I need a substrate to offer the cell some anchorage. Okay, so typically we can offer the cells some particles, as you see here in yellow. Not yellow, more orange here. Okay, another alternative would be aggregates. Okay, some stem cells can grow very effectively as aggregates. In the case of mesenchymal stromal cells, they do not grow very effectively in aggregates, but if you combine some biomaterials, namely those in the spherical shape, you can be quite effective in promoting attachment to these particles and then some proliferation afterwards. And then you have to combine these cells combined with the biomaterial with a specific bioreactor geometry. It can be a stirred vessel, okay, either mechanical agitated or with non-mechanical agitation, okay. But one of the mostly used systems has been this system here that you see at the center of this picture, which is the stirred tank. So over the years, we have been working hard on these microcarrier, these particle-based processes for the production of stem cells, mesenchymal stem cells in particular. Very recently, we have been focused hardly on using bioreactors that are single use. This means that they come disposable and ready to use, so they are more amenable to a clinical translation, okay, for therapeutic use. This specific bioreactor, as you can see in this image, has a particular way of mixing that is vertical, not horizontal, as most of the reactors that some of you might know, which has been described to cause minimal shear stress, which is really important for cells that are quite shear sensitive. And this comes available in different formats, okay? So it depends if you have the need of cells for 1 patient or for 100 patients, you have different volumetries to explore. Another aspect would be, okay, how can I get rid of animal components, the fetal bovine serum? So one of the opportunities that we had, it was to test a very interesting formulation of a platelet lysate, okay, using off-the-shelf platelets that were unused for transfusion that could be converted in another biomedical product this way to support the growth of stem cells in culture. So this is the so-called platelet lysate that has been used very effectively in clinical trials, especially in Europe, but more recently also in the US, in Asia. And basically the question was whether or not we can establish a bioprocess based on bioreactors combined with microcarriers, okay, to support mesenchymal stromal cell growth using this platelet lysate as an alternative to the fetal bovine serum. So what we did, we tested with different sources of mesenchymal stromal cells. We tested with umbilical cord, which is a very naive and very primitive source of these cells. Also adipose tissue, which is a very interesting source, less invasive compared to the bone marrow. We combine the culture with the platelet lysate, just as I mentioned, and also as I explained, these cells need to attach to some particles, so we did offer these spherical particles to the system, and they were kept agitated in this vertical agitated bioreactor, okay? So in these graphs, you can see that the cells grow quite effectively in the system. The filled lines are for umbilical cord. So umbilical cord cells, as more primitive cells, they grow faster compared to the adipose tissue. And interestingly, you can see also here some images of the cells. So here in blue, you can see the images of a staining that we use to cover the nuclei of intact cells, and we can see a gradual occupancy of the cells over time in culture. So these numbers are the time in culture, and we see more cells occupying the particles in the system. Okay. Importantly, if we can have a fancy system, but if it's too costly, it will be hard to be implemented at the clinical side. So one of the things that we had to demonstrate was the cost effectiveness of the process. And here in a side-by-side comparison, you can see for both cell types that we can reduce the manufacturing costs compared with the traditional manual method, which requires the flasks, okay, those Petri dishes or the flask that we use in culture. And here, as you can see, by comparing these black bars with the white bars, you can see that with the bioreactor system, we can decrease the manufacturing costs, okay? So at the end of the culture, we have to make sure that the cells do present the required characteristics in terms of identity, but also in terms of the potency. And this is really critical for mesenchymal stromal cells. Why? Because we have lots of different conditions and lots of different functions that can make more sense for one condition or another. So this is really important. So basically, most of the people that work with these cells, including our group, we have to demonstrate that these cells still differentiate into different cell types, namely bone, fats, and cartilage, and also if they identify some markers in terms of flow cytometry, and if they are functional in terms of supporting blood cells formation. Okay, so these are some of the potency readouts that we use in our lab. Okay, more recently, and quite aligned with the talk of Dr. Phillips that presented the way that senescent cells communicate through the secretory path that's secreting cells to the microenvironment. Recently, there has been this shift towards the so-called cell-free paradigm, whether or not we can replace very interesting cell therapeutics by the secretome that they produce. Okay, so not surprisingly, mesenchymal stromal cells appear as very interesting candidates to produce these secretome, namely these extracellular vesicles, including exosomes, among others. These extracellular vesicles have emerged as natural therapeutic agents, also as a drug delivery systems, and mesenchymal stromal cells, once again, are quite promising for the production of these therapeutic candidates. So for us, it was quite logical to, okay, if I have a good platform for the production of mesenchymal stromal cells, can I adapt the same platform for the production of the secretome, namely these extracellular vesicles? So you see, once again, the same type of approach, okay, this time also with bone marrow. So we tested the 3 different sources. We used exactly the same bioreactor system. And at the end of the culture, rather than the main focus being on the cells here in pink, the main focus was on these orange particles, the secretome produced by these cells, okay? The interesting part here, and this is a very popular and very competitive field of research for us, it was an opportunity to demonstrate what could be the advantage of using bioreactors for the production of the secretome. And here in this graph, you can see that the bioreactor performance is better than the static culture systems. But of course, you can also argue, okay, but you have a higher volume in the reactor. This is true. But then if you calculate a parameter that typically we can calculate, which is the production of these vesicles dividing by the number of cells that you have producing those vesicles, you can demonstrate that the bioreactor system can be quite promising in producing these therapeutics. And what we are doing now, so we are using these vesicles as we can use it as intrinsically a therapeutic agent, but specifically with a former student from precisely the biological engineering course, Miguel Fuzeta, we have been exploring the opportunity of using these vesicles for the delivery of peptides with therapeutic action, namely in the context of cancer. And this has been a new research line that we have been launching here at IBB in collaboration with my colleague Nuno Bernardes, that you also know. So this has been one of our focus, okay? And I think that without, I will jump into my conclusions because I do not want to delay the session. So basically what we can demonstrate is that for very interesting therapeutics as advanced therapy medicinal products, the routine methods for their production is quite conservative, okay? It does not explore all the technology that we have seen for other biologicals like the vaccines, okay? So bioreactor technology demonstrate to be quite valid and very promising for the production of these therapeutics, either the cells or their secretome. The single-use technologies are more amenable to clinical translation. And of course, we do have to do a side-by-side comparison with the bioequivalence of the cells and vesicles produced with the bioreactors versus the traditional systems, okay? So there is a long way to go since the time that we identify cells or vesicles with an interesting therapeutic potential towards the development of new therapies, okay? But that's why this is a research of engineers in collaboration with the biologists, in collaboration with the clinicians. And with this, I will end up. I will thank all the members of our team, all the funding, and thank you so much once again for the kind invitation. And I hope to have time at least for one question, okay? But thank you, Inês and André, for hosting this.
Thank you for joining us, and sorry that our time was a bit short.
Yeah, no, it's okay. It's okay.
I'm sure we'll find other chances to get back to this topic. Is there any question from the audience right now? Okay, I'm gonna start. I have one question. You know, based on your talk and all your experience, and I guess you've been doing this for a few years, so you can now appreciate the speed, how things are moving forward. Realistically speaking, how fast do you think this is going to be actually applicable, you know, in clinic? Because we've seen the results. There's like small cases here and there that show really promising, but then
yeah, the conventional and the thing in the United States, we still do not have the approval for these mesenchymal stromal cells, but it's on track. But in Europe, this happened, it's true. But then you take a look on the process part and it's quite limiting. And from my perspective, I think that I'm a big fan of the platform that we have, but I think that the microcarrier part, it's a little bit tricky because at the end, whether you have to release the cells from the particles in order to infuse to the patient, this is a critical safety issue. So I see with interest that moving towards the cell-free paradigm, it's more straightforward with the platform that we currently have because we couldn't, so we do not have the cells to be dissociated from the carrier. So this is, but still, I'm still a big fan of cells. I think that the secretome is quite useful, but I think that the cells need to sense the microenvironment to do a fine-tuning of what they are secreting. So I think that we still do not have all the knowledge to do this in vitro, to have the better secretome to produce then using a bioengineering approach. I think that this can be the future. But I think that lots of the limitations, and especially the current big prices that we have for this therapy is based on these very dummy ways of producing cells. Okay, so I think that we are really limiting in using very old-fashioned of producing these new therapeutics. Okay, and that's why I think that there is a window of opportunity for our cells engineers to innovate the way that these cells are produced.
Yeah, that makes sense. And then one day a company is gonna start, and then hope so. Okay, any other questions from the audience? Last chance.
I have a quick question. So I guess with the bioreactors, right? It's optimized for like cell growth. So do you see that there's differences, for instance, in the secretions if like a conventional culture versus the one that like the bioreactor where there's constant mixing? So if we were to correct for like cell number and everything, is the composition of those secretions like similar or very different?
What is described is that if you play with parameters such as the shear stress, the flow, the oxygen tension, for sure you can do a fine-tuning of what the cells are producing. So this is much more straightforward to do in a bioreactor with control. Okay, you have a control temperature, a control shear stress, a control oxygen levels. So for sure, I think, of course, I show you the simplistic part and the engineering part of the numbers, but there is also the other potential of turning these a more potent secretome with healthy cells that can promote regeneration, playing with the oxygen, the shear stress and other stimuli, even the addition of some chemical factors that you can induce the production. We have seen the production of classical cytokines and in the secretome, but for sure this is in our plans of exploring and do a snapshot of what is being different in static conditions compared with the bioreactor. But I think that there's lots of potential there.
Yeah, thanks.
Thank you. Thank you. It was a great talk also, and very nice to meet you, Jude.
Okay, thank you so much, everyone, especially for our speakers. Our next seminar will be October 29th. Put it on your planner. One of our speakers is going to be actually Nuno Bernard, who is here. And that's it. Stay tuned. Our website's going to be launched. See you all soon.
Thank you. Bye-bye.
Bye.
Bye.